and see the splendor, then the beauty that nature has to offer. But it's also a reminder to us that fall is here. It's just around the corner, and not long we'll see it here at the lower elevation. And I know a lot of us have probably finished harvest. Some of us are in the midst of harvest. And for a lot of us, we're in between harvest. We're waiting for that next crop to ripen. But now is a great time and a critical time, in my opinion, to think about next year's crop. One of the things that I talk about on this presentation today, and the title for this presentation is, what can we be doing now to set up next year's harvest? And so often in the fall, we look at building soil health. And Bruce used to talk that if we take care of our soils, the plant will take care of itself. But when we start to think about building soil health, we need to think, how does that affect our plant, and how does that affect overall plant health? And what we are doing right now to increase yield and increase plant health at the same time will build soil health. I so much look at when we talk about what dollars we spend in return on investment. And so often we think that what we're doing now and the investment we're making now, whether it be cover crops or biological inoculums or nutrition, how does that really affect our next season harvest? And that's really what I want to focus on today, how important now is, and how it, in the scheme of things, how this all builds to make next year's crop so much more successful. So when we start to talk about soil and plant health, when we look at the NRCS, the continued capacity of the soil to function as a vital living system that sustains plants and animals. And so often I look at that, if we read that backwards, plants and animals help sustain the living soil system. So it's all tied together. And when we start to talk about nutrient cycling and decomposition, and we think about that based on our soil, but our plants are critical in that of the nutrient cycling and the decomposition. But we also have to look at nutrient cycling and decomposition has to start now in order to ensure proper plant nutrient availability and soil health. We start to talk about how water infiltration is so key when we talk about building soil health. But I also think of, if we have the ability for infiltration of our water, we also have ability of root zones to penetrate deeper into that soil environment, basically accessing that water from that soil environment, but not only accessing the water, but mining nutrition much deeper in a broader profile within that soil environment. And same, we build microbial communities deeper within that soil environment. And we start to look at availability. I want to look a little bit about infiltration and how that affects the filtering and buffering. In so many places, whether it's Kansas, whether it's Nebraska, Colorado, Arizona, California, we're seeing more and more issues with high sodium levels within the soil environment. I'm seeing crops and soil analysis coming from all parts of the country where we didn't really used to have this problem. So when we start to talk about filtering, not only do I talk about holding nutrition, but also making it available so that we can leach like our sodiums out of our soil profile. And this only comes with good balanced soil structure, and the ability for that plant root to penetrate deeper into that soil environment. And the same holds true of buffering. I so often talk about millions of dollars are spent every year trying to address the pH. Everything we do right now in building soil health and building plant health will help make nutrition available, and it almost compensates for those excesses in pH, whether it's high or low, based on building that structure, that basically that environment to enhance that microbial community. And this comes through soil stability. As we build soil structures, as we build housing and homes for that microbial community, not only do we build soil structure, but we build filter and buffering, we build water holding capability, and we build nutrient cycling and decomposition. And that's the key, is we're building habitat for biodiversity. We so often talk, what does that microbial community need? It needs all of these things, and everything we can do right now is going to enhance the stability of our soil and build that for next year's crop. So both the plant and the soil will benefit from what we call the Fall Primer Program, and why it is so critically important to get started now. I put this slide up there. This is the research that is being done by Dr. Jerry Hatfield in Ames, Iowa. And this is so important because when we start to talk about adding biology, it increases all aspects of microbial response. And how we alter that biological activity within that soil environment is key to how we feed it. And we'll talk a little bit today based on crop-specific fall primer programs of what things do we need to do and think about of not only what biological inoculum are we going to use, but also what food are we going to supply to that to ensure that we have good microbial communities in addition to nutritional needs. We can never forget addressing the nutritional needs, not only for the plant, but also for that microbial community, because obviously that is a food source through digestion, making that and building that. It is also needed for that microbial community, and it's also needed based on when we talk about digestion of field debris within that soil environment. And Dr. Hatfield talks about how we can change or alter the soil aggregate response based on soil treatments, and that can happen in as little as 3 months for smaller particles and 9 months for the larger particle sizes. And the reason I throw this up here, because it all ties into why starting now with the fall program is so critical to overall yield increase and plant health, and not only building soil environment, but we're building an environment to grow a healthy plant. I always get asked the question is, what does my biological community, what do my beneficial microbes do in the fall, in the winter? All the research out there, for the most part up to this point, would say, well, they pretty much shut down. They don't do a whole lot. But I found this research years ago, and I thought it really applied right now to what we are going to talk about today. In Antarctica, basically researchers felt that blood falls came from a presence of algae in the water, and that's what gave it its red color. But on further research, they're noticing that it's because of the presence of salinity and iron that gives blood fall, the water, its colors, the red color. What is really important about this happened almost 2 million years ago, where a glacier sealed a small body of water containing microbes and covered it in a layer of ice. That microbial community metabolized the sulfate and the furic ions in order to survive. So it was food for them. They metabolized those as a food. They created the salinity and the iron that gives the blood falls its color. And all of this was done sealed by a glacier under a layer of ice. So when we start to talk about that, if they are undisturbed, they can still provide the function that they need within that environment, whatever that environment might be. So when we start to talk about seasonal microbial activity, and we look at like the early summer or the heat of the summer, who is active and functioning within that soil environment? So when we start to talk about the heat of Kansas or Nebraska or Phoenix, Arizona or California or any part of the country for the most part, we talk about, okay, when it's 100 degrees outside, who's going to be the most active? Well, in a lot of cases, it might be those stress reducers. They're designed to function within that environment. Generally, there's low moisture availability, high soil temperature, high heat, and the plant is under stress. It's calling out through root exudates to these stress reducers within that soil environment. So they're going to be our most dominant strain based on environment. When we start to look at late summer and early fall and the first frost, we start to see that some of those cellulose digesting bacteria are going to be most dominant. We're going to look at some of the solubilizers, the phosphate solubilizers, the nitrogen fixers. All of these communities are the ones that are going to be most dominant based on environment need and also root exudates going within that soil environment. So nature will dictate who is most active based on temperature and time and food, but they're all active even under cold, cooler temperatures, under frozen ground, deeper maybe within the soil profile. We're always going to have somebody who is actively functioning within that soil environment, just like the blood falls, sealed by a layer of ice by a glacier. So this is some information I want to get out there. What we do now with our microbial communities and how we feed them, they're still going to provide us a function within that soil environment.
When I was surprised, I saw some research a little while ago. You mentioned Antarctica. Actually, one of the soil organisms, it's normally a soil organism that we work with, the Arthropacter glommiformis, they found they're called cycrotrophic or cycrophilic, which are cold-loving versions of the same organism. They found that Arthropacter living under those Arctic ice sheets as well. And it's just so bizarre how nature finds ways to fill these different spots with these different organisms. Anytime there's food, anytime there's any sort of energy, nature's going to find a way to fill it with life. And it's just incredible how, like you're saying, Dennis, I mean, you expect winter, that soil's frozen. Well, as long as there's liquid water, there's going to be somebody active, moving, and doing some biological functions in there. It's incredible.
Well, and the part about that is I think the key to this is we talk about undisturbed. So they have a chance to create the environment with which they want to grow. And that's one of the things we're going to talk about today. So when we start to talk about, there's a lot of research out here, and we always refer to human health and soil health. So I talk a little bit about, in the mainstream media, we were talking about the effects of bacteria basically on moods, basically metabolizing food, controlling hormone levels, all the things that we talk about based on not only our soil environment, functions that beneficials can provide, but also we have to look at, there also research out there that members of a family, their pets who eat, breathe, and touch the same environment have same bacterial profiles. And the reason I point this out, this is one of the things that we talk about in a monoculture type scenario in agriculture. We start to really, in that environment, we create members of bacterial profiles or communities within that environment. And a lot of time that's based on what I go back to when we start to talk about the summer temperatures, the high heat. Who has become most dominant based on the environment that we have created, who is functioning within that soil environment. And that's one of the reasons why I talk about late fall, getting a good microbial community out there, creating diversity within that soil environment, and letting need of that environment dictate who is the most dominant strain. When we start to talk about that, we're going from an actively growing plant and roots to digestion, field debris, dead root mass, all of these types of things. We're shifting that community within that soil environment. So we almost have to basically re-inoculate that environment, get those players out there at a higher level so it doesn't take time to build those based on environment. We want to help that environment along. So I talk about that a little bit, and we always say that the microbial community within the soil environment basically needs food, water, oxygen, and shelter. And for the fall, I've added a couple things. So I want to talk a little bit about food, time, temperature, and water, because I think those are really critical when we start to think about fall soil applications, field digestion, field debris, overall plant health, putting plants into dormancy that are maybe those plants like our fruit trees, our nut trees. Anything that we can do, it's going to be a little bit different based on the crop that we're dealing with. So I say food is key. Building and maintaining a community within that soil environment is directly related to food. So how we feed these microbial communities and how we do this fall primer program is going to dictate what crop we're dealing with in the field. And I'll talk a little bit about crop-specific programs that we might want to look at, or things that we should probably be thinking about based on what is our crop and what are we trying to do. And the big thing is time. Fall is the longest period of time that we have in that soil environment that is undisturbed. So when we start to talk about an undisturbed environment, We give those microbial communities, like Steve was talking about, an environment in which they can build. We're not applying fertilizers. We're not applying fungicides. We're not disrupting. We're not tilling. We're not doing all of those things that disrupt microbial communities. And so when we look back at what Dr. Hatfield said, remember, we can change soil aggregate in as little as 3 months in the small particles or 9 months in the large particles. Well, in a lot of cases, if we're done with harvest now and we're not planting till the spring, we have almost 9 months to give that microbial community a chance to function within that soil environment. And this is one of the reasons why I feel the fall program is critical to build overall stability and plant health, digestion, nutrition availability. All of these things take time. And this gives us the longest period of time with temperatures. We're not in high heat. We talk about microbial community. What do they not like? High heat and direct sunlight. Well, our days are getting shorter, the intensity of our sun is not as great, and our temperatures are starting to come down, both soil temperatures and also the air temperature. How often have we been through the month of August where it's been so hot and we get that fall day in September and we sit out on our porch and we want to have a beer or an iced tea and just enjoy that environment that nature has provided for us? We feel like it's time to get active. We can get a lot of things done. Microbes feel the same way within that soil environment. As that temperature is reduced, as the days really get shorter, we can really see a lot happen based on digestion, based on nutrition availability, mineralization, all of those things that are so critical. And all of this takes water. When do we have the highest moisture content? Based on rain that stays within that soil environment. We have less loss to evaporation. Plants aren't utilizing as much of that moisture. So we maintain a more balanced moisture profile. And this is whether we're talking dry land or irrigation. So let's talk a little bit about crop specifics and how we might feed those. So, obviously there's a lot of information out there about cover crops. Obviously, cover crops are a great way to build soil health and help make nutrition available to that plant for the long-term coming up cash crop. But when we start to talk about that, we look at the field debris here on the right, and Dr. Hatfield refers this as an active growing or a passively growing cover crop. Both of these are cover crops, and both of these we can achieve the same thing based on overall nutrition, digestion, and plant health. But it's done a little bit differently. This picture you see on the left, this is a grower in California that sent me pictures of his cover crop. He's so excited because of the amount of root mass and the microbial community that he is seeing growing on this plant, understanding what he is doing to build that soil health. And over here on the left, we see cover crop. This is from the NRCS out of Idaho. And there's some really good information here based on the number of bacteria increases from 5 to 2,000 times with a cover crop based on just regular soil. And I think a lot of that has to do with whether it's 5 times or 2,000 times of what are we doing when we plant that cover crop. If we just put that cover crop in the ground and we don't think about our microbial community, we don't think about inoculation, we don't think about what food does that cover crop need in order to survive, that plant cannot function at its full genetic potential. And if it can't do that, whether we're talking yield on a tree to take to market or we're talking yield within our soil environment, we cannot get the same yield as we could if we pay attention to those types of things. So we have to look at this. What are we trying to do and how can we help our systems along when it comes to building soil health? A lot of times we talk about sugars and feed me. I put this in there because it's all about feeding that microbial community within that soil environment and also feeding that crop. In some instances, I feel sugar is good. We'll talk about that a little bit. When we have an actively growing cover crop and it is putting the root exudates, it's putting those sugars, those proteins back into that soil environment. Is sugar as needed? I don't think so. But as we get into the late fall, we'll talk about here a little bit, on a cornfield that's harvested in late October or early November, temperatures are a little bit cooler, you know, a little bit of molasses might just help to feed that microbial community, get it up to population, get it a little bit more active, and get it functioning within that soil environment. But the benefits of not only a cover crop, and I call cover crop, that can be a living plant, that can be field debris, and we'll even talk about how to build soil structure and nutrition availability in bare soil, which is our least favorite, but we can still do it.
So Dennis, you have those pictures of dreadlocks there, and I mean, that's, earlier you referred to Dr. Hatfield's research in showing that, you know, 3 to 9 month span of increasing soil aggregate formation. Those dreadlocks, those are the micro aggregates that are forming, I mean, almost on a daily basis. We've seen examples in the field where we're seeing that in even shorter amount of time. I mean, when you go out into a greenhouse and you have fresh starts, or you have a plant that's in the field that's recently been inoculated, we've seen massive shifts in matters of weeks with the amount of that micro aggregate, those dreadlock formations that are happening. So it's pretty incredible. And so when you say that some of these cover crops only have 5 times higher population densities than regular soils, it just makes me laugh, because I'm trying to figure out, well, what are those guys doing so that it's only 5? It just doesn't make sense to me.
Well, and so much of that is, I agree, Steve. We cannot look at a cover crop of the wheat, throw that out here. I look in the Washington area, dry land where we're very limited on moisture, especially in the fall. Generally, wheat crops come off, depending on what part of the state you're in. But, you know, usually we're done in September with most of the cutting of the wheat. Whether, you know, there's some spring wheat maybe still out there that needs to be harvested. But if we pay attention to the need, I believe that there is a possibility of doing cover crops in parts of Washington state. We may not be able to get it done every year. Maybe it's every 3rd year. So we have to look at that. But, you know, I look at, as I go on and as I learn more about what we can do, and especially with the fall being critical in order to build all of these types of things, is I'm seeing an actively growing plant is a wonderful thing to have, but in some environments it's just not possible. So how do we work with what nature has given us, and how can we build that same thing? And there's ways to do it, and that's what we need to focus on. You know, Steve's talking about seeing dreadlocks created on the roots of different plants in the greenhouse or in a very short period of time. But what I am finding, again, this picture here on the left is rye cover crop. This was in Kansas. You can see the microbial community, the aggregate that we are forming around that root zone. But what we're finding is, whether we do it with a cover crop or whether we're doing it with a passive field digestion of debris, we are seeing the same inoculation on the plant in the springtime when we plant that cash crop into that soil environment. You know, John Kemp talks a lot with AEA a lot about the genetic potential of a seed, and what is that potential, and everything we do to reduce stress off that seed is going to increase yield. When we start to see these microbial communities inoculate that root, they're providing nutrition to that plant, they're digesting, they're eliminating stress off that plant. So the earlier we see that form on the plant, the better chance we have of higher yields and increased yields for our crop. And I'm seeing this all the time now, where we see pictures like you see here on the right-hand side of inoculation of plants with just beautiful microbial colonies inoculated right around that root zone. So when we start to talk about a little bit, how do we do this in bare soil, field debris digestion, all of these types of things? So when we start to think about corn can leave 3 to 4 tons of surface residue per acre and 1 to 2 tons of root biomass. That is a lot of carbon. And so how do we digest that? In a lot of cases, this is where we recommend the biodigester with the food source based on crop residue. And the reason I say that is that's very important, because we need to make sure there's a nitrogen component within that in order to break that carbon chain. A lot of the growers that I talk to now are very concerned about digesting too quickly. We see with biodigesters and good nitrogen levels, we can almost digest that field debris quicker than they would like over the winter time. They want to leave some of that debris on the soil surface as a protection from rain, from wind, from erosion, all of those types of things. So we need to pay attention is how quickly do we want to digest our field debris. So if we're looking at soybeans, which generate notoriously very little surface residue and not a lot of root residue within the, or root biomass within that soil environment, you know, biodigester may not be the best choice. Maybe a spectrum application with a little bit of fish and humates, because obviously we don't have as much carbon, so we need to support that microbial community. We need to give it more food. So let's throw some humates out there, maybe a little bit of sugar if it's late fall application, and make sure we're feeding that community to get those populations up. So we have to pay attention to what are we digesting and how are we digesting it. In the bottom right picture here, you see one of the wheat growers. We have less carbon to deal with. So again, how quickly do we want to digest that within that soil profile? I talked to a grower in Nebraska this year. In their crop rotation, triticale was followed by corn, and they noticed that there was actually a nitrogen deficiency in their corn early on after the triticale, because we did not digest that stubble within that field environment enough in the fall. So what they did, they had applied it biology and nitrogen in the spring. Well, it actively got that digestion process going. But in the short term, it tied up a little bit of nitrogen for that digestion. So these are the things when we start to talk about what can we do now this fall in order to build overall plant health and soil health are so critical.
So do you think that farmer, if they had their triticale and they were working on it earlier in the season, they wouldn't have seen as much of that tie-up?
That is some of what we talk about, okay, is digesting that early based on the amount of stubble. Now that may not be every year. Some years where we have higher spring moistures, we have more field debris, we want to come in with the biodigester and a little bit more nitrogen. On years of maybe not as good as spring and less field debris based on stubble, based on crop residue, then we have to think about, well, we may want to back off. How are we going to, what are we trying to digest and how quickly do we want to digest it? So when we start to talk about, we're talking about the short-term tie-up of nitrogen. But the key to this in the long term, we increase nitrogen. So again, this goes back to the idea of we have the longest period of time with undisturbed soil to make a difference. So that's exactly what we just talked about. More time to digest, to get that mineralization going, to get that nutrient cycling going is going to pay dividends in that plant the next spring. Carbon will remineralize our soil with microbial decomposition. That's what we're talking about, but we need time in order to do that. We'll change the texture and the quality. And the big thing here is we can reduce crusting and compaction. This is actually also coming from Dr. Hatfield, some of the research he's doing. We're now seeing it with the NRCS. It's something that we have talked about for years, is as we build soil structure, as we have those glomulins, those polysaccharides, those glues, basically forming soil aggregate, we start to see a reduction in compaction and crusting. And that is so important based on what I talked about, and I'm going to talk about a little bit more here in a minute, based on sodium issues. And then we talk about it holds macronutrients. It holds the zinc, the manganese, the copper, the boron. It builds CEC. It helps us with potassium, magnesium, calcium. It builds humus. It builds organic matter. All of these things are exactly what we need to be doing right now to ensure plant health. So we talk about digestion and why is that so important on our field debris. And the example I like to give, this is a test that Steve had done based on compost. And it was 2 sets of compost. One was the control that came out at 74 million colony forming units, and then inoculated with biogenesis. The compost treated had 4.4 billion colony forming units. So as we talk about with a cover crop, and we saw that 5 to 2,000, we can see those same types of things when we start to address the need of our field debris to break it down within the soil environment. And the example I give here all the time is, I don't care if you have 1 acre or 300 acres or 600 acres, let's make a 600-acre compost pile. Let's digest that compost in the field. Let's mineralize that, build soil structure. And the way we do that is how we feed it and how we treat it. An example of this is a grower, persimmons and kiwi in California that Steve and I visited this last, or I guess early this spring. Yeah. What we found so interesting is this picture on the upper left. You can see the grayish soil that is outside of the growing area of the trees. And then you go inside that and you see where the field debris is left. You see a little bit different color. On the right here, you can see the picture of the soil that is where the field debris is being left and a handful of that sandy native soil. What we saw, which was so interesting to look at, and a bottom right picture, you can kind of see the difference between the two, is everywhere in that field where they had good activity, we were breaking down that field debris. We did not have any standing water. You could walk in the low areas, in the high areas, everywhere we went, you could see that the water was, we had good infiltration rate throughout that soil environment. And yet you can look right outside that environment, next to that pole, we have standing water. You go down here into a low area, you dig down into that soil, it smelled like sewer sludge, it was anaerobic. Nothing will grow here, but you can see there's no debris on top of that soil. All that green you see growing there is not really plants, it was mainly algae that was growing on top of that soil. And you go right away and we have good, healthy smelling, sweet smelling soil. It's all based on digestion. And this is just a prime example of what can happen. So I talked a little bit about high sodium. You can see this picture here on the right. We're seeing more and more of this, especially under drip irrigation. We see that crusting of that soil. A lot of this is caused because of the high sodium content. We have created an environment within this, around this soil, around this plant, where we're concentrating high levels of sodium through irrigation water, through salt-based fertilizers into this environment. Right now, the only way to reduce that is to do a little bit of tillage, lightly break that up. You can see the, I guess, the dirt clods around those plants. This is what we have to do in order to get water penetration within that. But if you think about that, in June, July, August, we are not building microbial communities within that soil environment. We have sealed off that soil. The temperature is too hot. It's too dry. So anything we can do in the fall, right now, I would recommend the Spectrum DS anytime we have high sodium content within that soil environment, and add a little compost, as long as we pay attention to our compost to make sure it's also not high in sodium that we're applying back. Steve and I saw one the other day that I would think it was 3,000 parts per million sodium. We do not want to be applying that to our field. That is not good compost. We need to add humates and calcium anytime we have a high salt issue within a soil environment. Let that DS work. And again, we have time on our side. Give a chance to, we have high moisture, we have rain, we have spring moisture. Let's try and leach some of that out of the soil profile to create a better environment for that plant. If we can put a wheel line out there, if we have pivot irrigation, let's try and leach some of that through after we've applied our Spectrum, our humates, our calcium. Let's try and break some of that up, break that bond, get that to leach through that soil profile and make it so we have a better growing environment. You can see on the right how stressed this plant is. If we can put a cover crop in there, that's going to help out also. So we just have to pay attention. If we have high salt irrigation water, I think we're still okay to leach some of that out if we're in a drip type scenario. If we're not in a drip type scenario, we may question whether running a lot of irrigation water and adding to the problem is the way to do it, or let Mother Nature take her course.
So one thing I would add there, you mentioned the calcium, you mentioned the humates. If you can get a little bit of kelp, some of the Cytogreen or Micro 5000 organic or some other kelp materials, kelp, the phytohormones and some of the minerals in there can definitely help with the plant stress as well as those beneficial spectrum DS organisms.
You know, and when we get crusting on our soil, Steve, how does that affect our microbial community?
Well, I mean, Dr. Hatfield talked about 1/32 of an inch is enough to stop all gas exchange. We're working with an aerobic environment, hopefully. And if we have basically any visible crusting at all and we have stopped gas exchange, we have shut down the organisms that we want to be growing there. Earlier, you mentioned the idea of the people, the animals, all these things that are in the same environment, that are touching the same things, eating the same things, end up with kind of a similar microbiome. And you also mentioned how the microbiome is really important for the mood of the family. The mood of these plants is kind of the same thing. As we seal off and crust off that soil and we change those populations, you can kind of think about the mood of the plant based on the microbiome around its roots is going to change as well. That plant is crying out for help. So like you said, a little bit of field debris in there, some compost, some lights, disking or some lights breaking up of that crust to help the gas exchange is going to make it so that our spectrum organisms have a chance of reestablishing with that plant.
Yeah, thanks for that good information. And this same's true, you see this a lot, is coming along with the hemp crop, which is taking off across the country. You can see here the picture on the right. This is one of my growers in Colorado. I put this in there because, as you see, in between the rows are peach trees. And then in between those rows are hemp plants. So I've talked about this before. This is generally when we break a new field and get ready to plant, or we take out existing trees and get ready to put a new crop in. Our first rotation of that is watermelons to build soil health. Mainly that's what we're doing, but we get a little income off of the watermelons for that, for the grower. So he'll take and create an environment. He'll bring in some compost. He'll open up that soil. He'll get some spectrum in there, or spectrum DS if we have a high sodium issue. We'll grow watermelons for a year, addressing all the nutritions. And then the next year we come in, we root dip the plants, and we put them into the soil environment. Now in between those rows, while we're waiting for that second or third year to get a cash crop off of these, in this case it was peach trees, we're growing hemp in between each of those rows. This is what the grower is doing. And so under plastic, we are planting with drip irrigation, inoculating with biology. And he was talking to me the other day. We haven't even harvested this crop yet, but he's already understanding what things do I need to do this fall to compensate for what I've done here in this row with my hemp plants, based on knowing that we probably had a little bit lower oxygen level with gas exchange with the plastic. We probably concentrated a little bit of sodium within that environment. So he's already understanding he needs to remove the plastic, he needs to add the fall primer and nutrition, use his Spectrum DS, he'll put some compost in there if needed, he'll incorporate and he'll water, and he'll let that digest over the winter time. So this is all really important. We need to be thinking now, this is the reason I point this out, of what we can do as soon as that crop comes out of the field, because a lot of times we're limited on time. One of the things I always like about the fall program, and what really makes the fall primer program so attractive, is it gives us an opportunity where generally we have the best weather, we have equipment available, we have people available to get all of these things done. In the springtime, Mother Nature does not always play fair with us. So if we can't get into the field because of excessive moisture in the field, we can't get equipment in there, there's other things that we're spraying at that time of year. The closer we get to planting, or that plant or tree coming out of dormancy, the less time we have for this microbial community to make its difference within that soil environment.
What I like, you mentioned there that, labor, getting help. If you're in the middle of harvest, trying to get some extra people out there to do some of this, it's just not going to happen. You mentioned that fall, after we've done our harvest, we've usually got those extra help. We've got those man hours that we can get some people out there really paying attention. I really like that point, Dennis.
Yeah, it's a great time to get things done, and the weather is usually
comfortable.
Yeah, it plays fair with us at that time. And my favorite, obviously, is actively growing fruit trees. This is one of the crops I absolutely love to work with. We have the longest period of time to make a significantly difference, not only on plant health, but also soil health. We can build soil health over years rather than trying to do it over the short term. One of the things that I look at is we need to combine, anytime we look at fruit trees, foliar nutrition with soil inoculation. They work hand in hand when we start to talk about root exudates. In most cases, we're not talking about a lot of field debris. So generally, I am talking about the Spectrum or the Spectrum DS at this time of year with the Nutrinede, or the Pepzyme G, depending on whether you're organic or conventional, and then going with the foliar nutrition program based on need. One of the things that I look at here that I talked to a grower, as soon as we are done harvesting that fruit, we need to come back with an irrigation cycle. We need to get the stress off those plants because generally we've dried out the ground and we've stressed the trees a little bit by picking the fruit. But we need to come back in immediately with the soil inoculation first, followed by a foliar application following. And a lot of time that is based on a tissue or plant sap analysis, and we're going to start to do corrections within that for that tree. We have already set next year's fruit. So everything that we can do for that plant after harvest is building next year's crop. It's kind of interesting because there's a spray tech at one of the orchards. He's been with the orchard for about 20 years. The owner that I've worked with forever took that orchard over about 12 years ago. And I had talked to the spray tech about why this is so important. And the first couple of years, you know, he was kind of like, you know, that's okay. You know, I'll kind of do it. But as time went on, he is visually seeing the difference in the plants. I think about 4 or 5 years after that I was there, one of the things that he had made the comment of the orchard is alive. The orchard has never felt this way before. These are the things we start to do start to see when we start to build this fall program into what we're already doing. And we refer to that as with this one grower as the pack house problem. He was telling me this the other day, is when he brings a semi full of fruit into the pack house, they refer to him as the pack house problem. He always brings in the largest fruit. It makes all the other fruit basically within the pack house look inferior, and they have to change everything they're doing. The way they handle it, it's larger fruit. So how do we get there? How do we consistently bring in quality fruit to the pack house? And how do we make sure that we ensure pack out of all the fruit that we bring? And so often it's going just beyond a tissue and a soil sample and a plant sap analysis. It's looking at our orchard, looking at the leaf structure, looking at the size of the leaf. You know, if you go back here to this picture, I don't know how well you can see it, but if you look at the leaf size here, these are cherry leaves. And as you can see, these cherry leaves are the size of the palm of your hand. These are huge leaves. They're very symmetrical. They're well grown. They have good color. We have good overall plant health. We maintain that through the entire lifespan, the entire crop and growing year of that tree, but also the entire lifespan of that tree. And so we get consistently high yields off of that fruit based on nutrition. So the way we do that is overall plant health. How do we see that? Walking through the orchard is great. I recommend a drone to fly above so we can get an aerial view, or get on a high perch and look down on the orchard so we can see the inconsistencies of our plants, basically from a bird's eye view. Check the roots, understand what our roots are doing. Obviously, we can't dig up a fruit tree and look at the roots of what it's doing like we can corn, but walk through and have an understanding based on soil structure, based on where we might have compaction. What are our roots doing and where do we need to feed that tree based on a nutrient being applied to the soil? Smell the soil, look at the soil, dig in the soil, let the soil tell us what it needs and what it is doing, this will all give us better indicators of overall plant health.
If you've got the time, the Graham Shepard Visual Soil Assessment Volume 3 is a great way to spend an hour or so in your field, kind of digging around and looking at the modeling, looking at your worm counts, looking at your aggregate formation. It's an additional tool to help you kind of evaluate your soil quality.
So with that being said, fall is a great time to really see what our plants are telling us. Take a look at them. And I always recommend test, don't guess. One of the things I look at is when I see fruit trees, I love to have a tissue sample or a plant sap analysis off this tree right now. And the reason for that is it indicates what do we need to be foliar feeding now to ensure a healthy crop for next year, but it gives us a crystal ball. What we can do next year in the springtime, we know what our deficiencies were going into the fall. So it gives us an idea of what we need to feed based on the spring. Now, when we say that, is we also need to look at plant need. What nutrition does it need early in the spring? If we have a potassium deficiency, we don't want to be applying a lot of potassium in the springtime. But if we have trace minerals, if we were a little deficient on nitrogen, it gives us indicators of what we need to be applying in the springtime. Because if you think about it, in the springtime, so often that plant comes out of dormancy. We want to give it a foliar package, but the leaves aren't mature enough yet to pull up plant sap or a tissue analysis. We have to wait. If we have a fall tissue analysis, we know immediately, or we have an idea, what does that plant need? Let's get that out early while we're waiting for those leaves to mature, and then let's pull another tissue analysis or plant sap, and then continue our foliar program based on that information. So it's our crystal ball. It tells us what we need to do this fall and what we should be planning for the next spring. And the same has to do with a soil analysis. What is our soil telling us? Get an analysis now. We can put down soil applications in the fall. Again, what do we talk about? Time. Let that microbial community digest that, build that. And so when we plant that crop in the springtime, all of that is available to the plant in a state the plant can use it. We don't have any kind of lag based on temperature or waiting for that microbial community temperatures to get right to come up at a high enough population to make enough of it available. They've already done some of their work. They've stored it, they've put it in the shed, it's in the freezer. So when they come home from work late at night, you take it out and you pop it in the microwave, I guess. Although I don't like microwaves, but I mean, it's kind of the same idea. Let's store that into our food cupboard. Let's make it available to the plant so when it comes out next spring, it's already available. One of the things I always tell growers at the end of the season, now is a time to evaluate yourself, evaluate your program, evaluate the results, and share that information with your crop advisor, with your distributor. Give those individuals the same information. Let them know what you're trying to work on, what went well, what didn't go well, what we need to work on for the next year. The orchard you see here in the background, that's one of the things we did every year. When he first took this orchard, we had a huge list of things that we paid attention to, that were critical, that he wanted to get done. Now the list is much smaller, but we always have something that we want to focus on that we can do better within that orchard to help create higher productivity, higher yields, and better profits for the grower. And the only way we can do that is if you share that information with your, like you say, your crop advisor, share it with your distributor. I say share it with as many people as you can, get as much information as you can, then you, the grower, can make a determination of what best fits into your farm. So with that, I guess we'll go to questions. Thanks for everybody. If you have any questions, please let's go ahead and ask them now.
Oh, thank you, Dennis. Are there a couple up there? Click on the chat and see if you can see anything. Hello to Germany.
Okay.
This is Jenny here. Just wanted to let you know I'm already getting requests for the link to the audio for today. There's a lot of good information people want to listen to again. Well done.
Excellent.
Okay.
So yeah, so if anybody has any questions pertaining to what can I be doing now based on fall primer applications. What things are important? What didn't I think about? What should I be thinking about? Feel free to contact us here in the office. Myself, Steve, Mark, will be more than happy. Send us an email, whatever we can do. We look forward to working with you this fall. And I want to thank everybody for attending, and we'll talk to you soon.
Thank you, everybody.
One other thing I guess I will tell you is next September webinar. That's the third week of September. John Kemp with AEA is going to be attending with us. We're going to be doing a roundtable. So we'll continue on this topic, specifically about how microbes, I believe, address this nutrient availability. So please attend in September. We'll look forward to seeing you. Thank you.
Thanks, guys. Well done.
Thanks, Jenny.